Welcome to our channel Medicine A to Z. Today we are going to discuss physiology of blood pressure regulation. Blood pressure is the pressure of blood pushing against the walls of your arteries. Understanding blood pressure involves grasping the relationship between cardiac output and total peripheral resistance. Cardiac output, the amount of blood pumped into circulation per minute. Total peripheral resistance, TPR, refers to the overall resistance to blood flow in the systemic circulation. Cardiac output is calculated as the product of stroke volume and heart rate. Stroke volume represents the amount of blood ejected from the left ventricle with each contraction, while heart rate is the number of contractions per minute. Stroke volume, a crucial component of blood pressure regulation, is influenced by several factors. Stroke volume, abbreviated as SV, represents the volume of blood ejected from the left ventricle during each contraction. Preload refers to the amount of blood in the ventricle at the end of diastole, just before the next contraction, which determine the initial stretching of the heart muscle before contraction. An increase in preload typically leads to a greater volume of blood ejected with each heartbeat. Contractility refers to the forcefulness of cardiac muscle contraction. Enhanced contractility results in a more vigorous ejection of blood, contributing to increased stroke volume. Afterload represents the resistance the heart must overcome to eject blood into the systemic circulation. A decrease in afterload facilitates more efficient ejection of blood and consequently enhances stroke volume. Now, let's delve deeper into one of the fundamental factors influencing stroke volume. Preload. Preload refers to the amount of blood in the ventricle at the end of diastole, just before the next contraction. It's determined by factors such as venous return and the volume status of the individual. Essentially, preload is the degree of stretch of the ventricular muscle fibers just before contraction, which affects the force of the subsequent contraction. When blood volume increases, preload typically increases, leading to a stronger contraction of the heart. Conversely, a decrease in blood volume usually leads to a decrease in preload and consequently a weaker contraction. Now, let's delve deeper into contractility. The contractility of the heart refers to its ability to contract effectively in response to electrical stimulation, independent of changes in preload or afterload. In simpler terms, it's the strength and vigor with which the heart muscle contracts during each heartbeat. What is afterload? Afterload refers to the resistance that the heart must overcome to eject blood out of the left ventricle and into the systemic circulation. In simpler terms, it's the pressure that the heart must generate to open the aortic valve and push blood into the aorta and the rest of the body. Afterload primarily depends on the systemic vascular resistance, which is the resistance to blood flow offered by the arteries throughout the body. Heart rate, another component of blood pressure regulation, is under the influence of various factors. Let's explore these factors and how they modulate heart rate. Sympathetic and parasympathetic nerves, part of the autonomic nervous system, play opposing roles in regulating heart rate. Sympathetic stimulation increases heart rate by releasing neurotransmitters like adrenaline and noradrenaline, which bind to receptors on the heart muscle cells, stimulating them to beat faster. In contrast, parasympathetic stimulation, mediated by the vagus nerve, decreases heart rate by releasing acetylcholine, which slows down the heart's pacemaker activity. Ions such as calcium, potassium, and sodium are essential for the generation and propagation of electrical impulses in the heart, influencing heart rate. Hormones like thyroxine, adrenaline, noradrenaline, and cortisol can directly or indirectly affect heart rate through their actions on cardiac muscle cells or the autonomic nervous system. Total peripheral resistance, or TPR, plays a pivotal role in regulating blood pressure by determining the resistance to blood flow in the systemic circulation. Let's explore these factors and their impact on total peripheral resistance. Firstly, TPR is influenced by the length of the blood vessels. Longer blood vessels offer more resistance to blood flow compared to shorter ones. This happens when you are obese, you total blood flow. Length increases leading to hypertension. Secondly, TPR is affected by blood viscosity, which refers to the thickness or stickiness of the blood. Higher viscosity increases resistance to blood flow. Dehydration and polycythemia are two good examples. Lastly, TPR is inversely proportional to the fourth power of blood vessel radius. This means that even small changes in blood vessel diameter have a significant impact on resistance. Therefore, the degree of constriction or dilation of blood vessels can dynamically alter TPR. Increased constriction leads to higher resistance, while dilation decreases resistance. In summary, blood pressure regulation is influenced by several key factors. Preload, contractility, and afterload directly impact stroke volume, a determinant of blood pressure. Sympathetic and parasympathetic supply, ions, and hormones modulate heart rate, another component of blood pressure. Total peripheral resistance, influenced by the length of blood vessels, blood viscosity, and blood vessel diameter, regulates blood flow in the systemic circulation, thereby affecting blood pressure. Physiological blood pressure regulation. Blood pressure regulation involves a sophisticated interplay of physiological pathways. Baroreceptor pathway, Renin-Angiotensin pathway, ADH pathway, ANP pathway. The baroreceptor pathway plays a crucial role in regulating blood pressure by detecting changes in blood pressure and relaying signals to the brain. Baroreceptors are specialized nerve endings located in the carotid bodies and aortic arch. These baroreceptors contain mechanical sodium channels. When blood pressure increases, these channels open, allowing more sodium influx into the receptor. Once the sodium influx reaches a threshold, it triggers the generation of an action potential in the baroreceptor. The carotid sinus baroreceptors transmit their signals via the 9th cranial nerve to the tractus solitarius nucleus in the medulla of the midbrain. Similarly, the aortic bodies transmit their signals through the 10th cranial nerve to the tractus solitarius nucleus. In response to these signals, the brain adjusts heart rate and blood vessel diameter to maintain blood pressure within a narrow range. The baroreceptor pathway is a vital mechanism for regulating blood pressure, involving a complex network of sensory and motor signals. Afferent signals from the carotid sinus baroreceptors travel via the 9th cranial nerve and from the aortic bodies via the 10th cranial nerve to the tractus solitarius nucleus. In the tractus solitarius nucleus, these sensory signals are integrated to assess blood pressure status. Efferent parasympathetic signals travel via the vagus nerve to modulate heart rate and vessel dilation, while sympathetic signals travel via descending fibers to the lateral horn of the spinal cord. In the lateral horn, sympathetic signals, synapse, and postsynaptic fibers reach the heart via sympathetic ganglia, leading to adjustments in heart rate. The baroreceptor pathway orchestrates precise adjustments in cardiovascular function to maintain blood pressure within a narrow range, ensuring optimal tissue perfusion and overall physiological balance. When blood pressure increases, baroreceptors in the carotid sinus and aortic arch are stimulated. These signals are transmitted to the nucleus tractus solitarius in the brainstem via the 9th cranial nerve from the carotid sinus and via the vagus nerve from the aortic arch. In the nucleus tractus solitarius, these signals inhibit the descending efferent sympathetic fibers, leading to relaxation of vessel walls and a subsequent reduction in blood pressure. The inhibition of sympathetic outflow results in a decrease in heart rate and reduces the release of epinephrine and norepinephrine from the adrenal medulla. Additionally, sympathetic inhibition reduces renin secretion from the juxtaglomerular apparatus, further dampening the renin-angiotensin-aldosterone pathway. The reduction in sympathetic supply causes vasodilation of afferent and efferent arterioles in the kidneys, increasing blood flow and urine production, ultimately reducing blood volume. Additionally, the baroreceptor pathway stimulates parasympathetic supply to the heart via the vagus nerve, further reducing heart rate. Renin-Angiotensin-Aldosterone Mechanism. The juxtaglomerular apparatus is a specialized structure located in the kidney where the afferent arteriole and distal convoluted tubule come into close proximity. The juxtaglomerular apparatus consists of three main components. Macula densa. The macula densa is a group of specialized cells in the wall of the distal convoluted tubule. It is in direct contact with the arterioles of the renal corpuscle. The macula densa cells monitor the concentration of sodium chloride in the tubular fluid. When the sodium chloride concentration is low, it signals for renin release. Juxtaglomerular cells. Juxtaglomerular cells are specialized smooth muscle cells found in the walls of the afferent arteriole. These cells secrete the enzyme renin in response to signals from the macula densa or other regulatory mechanisms. extraglomerular mesangial cells. These cells are located between the macula densa and the arterioles. They are thought to play a supportive role in the function of the juxtaglomerular apparatus, though their precise function is still being elucidated. The Renin-Angiotensin-Aldosterone Mechanism is a vital regulatory pathway involved in maintaining blood pressure and fluid balance. When blood pressure decreases, filtration through the Bowman's capsule also reduces, leading to decreased sodium levels in the distal convoluted tubules. The macula densa, sensing this change, signals the juxtaglomerular cells to produce renin. Renin, an enzyme released by the juxtaglomerular cells, cleaves angiotensinogen to form angiotensin I. Angiotensin I is then converted to angiotensin II by endothelial cells in the lung microcirculation. Angiotensin II exerts numerous effects, including vasoconstriction, which increases venous return and cardiac output. It also causes arteriolar constriction, leading to increased total peripheral resistance and subsequent elevation of blood pressure. Angiotensin II stimulates the synthesis of epinephrine and norepinephrine from the adrenal medulla, further enhancing vasoconstriction and increasing blood pressure. Additionally, angiotensin II directly stimulates the hypothalamus, leading to increased thirst sensation and secretion of ADH, which increases blood volume and pressure. Furthermore, angiotensin II stimulates sympathetic nerve endings, increasing sympathetic drive and further promoting vasoconstriction. Lastly, angiotensin II causes vasoconstriction of both afferent and efferent arterioles in the kidneys, reducing blood flow and urine production. ADH mechanism. ADH, or antidiuretic hormone, or vasopressin, is formed in hypothalamus and transported to posterior pituitary via axons and secreted to blood. ADH acts via V1 receptors on blood vessels and causes vasoconstriction, then increase peripheral resistance and increase BP. It also acts on distal, convoluted tubules and collecting ducts, and increase the expression of aquaporine channels, and finally increase water reabsorption. The antidiuretic hormone, ADH pathway, is another crucial mechanism involved in regulating blood pressure and fluid balance. When blood pressure is low, baroreceptor stimulation decreases, leading to a reduction in impulses from the tractus solitarius to the hypothalamus. When the inhibition is removed, ADH production increases in the hypothalamus. Additionally, angiotensin II directly stimulates the hypothalamus to produce ADH. Increased osmolality and elevated sympathetic drive also contribute to increased ADH production from the hypothalamus. ADH acts on the distal convoluted tubules to increase water reabsorption, conserving water and increasing blood volume. Furthermore, ADH causes vasoconstriction, leading to increased peripheral resistance and subsequent elevation of blood pressure. ANP pathway. ANP, or Atrial Natriuretic Peptide, is from atrial wall due to stretch of the wall. Promote vasodilation, which helps to reduce systemic vascular resistance and improve blood flow to tissues. Enhance sodium and water excretion by the kidneys, which helps to decrease blood volume and preload on the heart. Inhibit the Renin-Angiotensin-Aldosterone System, which can reduce vasoconstriction and sodium retention, further aiding in blood pressure regulation. Atrial Natriuretic Peptide, ANP, plays a vital role in regulating blood pressure through its multifaceted functions. When blood pressure increases due to elevated fluid volume, the arterial walls stretch, triggering the release of ANP into the circulation. ANP exerts three primary functions to help reduce blood pressure and maintain cardiovascular homeostasis. Vasodilation. ANP causes vasodilation, leading to relaxation of blood vessel walls and a subsequent decrease in blood pressure. Inhibition of the Renin-Angiotensin-Aldosterone Mechanism. ANP inhibits the Renin-Angiotensin-Aldosterone Mechanism, which helps to prevent vasoconstriction and sodium retention, contributing to blood pressure reduction. Inhibition of water and sodium reabsorption. ANP inhibits water and sodium reabsorption from the distal convoluted tubules in the kidneys, promoting diuresis and natriuresis, which further reduces blood volume and blood pressure. In summary, ANP acts through vasodilation, inhibition of the Renin-Angiotensin-Aldosterone mechanism, and inhibition of water and sodium reabsorption to help reduce blood pressure and maintain cardiovascular balance. ANP is more important in heart failure in reducing fluid overload than in blood pressure regulation. Blood pressure regulation involves a complex interplay of various physiological pathways aimed at maintaining hemodynamic stability. Key mechanisms include baroreceptor pathway. Baroreceptors located in the carotid sinus and aortic arch detect changes in blood pressure. When blood pressure increases, baroreceptors signal to the brain to decrease sympathetic activity and increase parasympathetic activity, leading to vasodilation and decreased heart rate, ultimately reducing blood pressure. Conversely, when blood pressure decreases, baroreceptors signal for increased sympathetic activity and decreased parasympathetic activity, leading to vasoconstriction and increased heart rate to raise blood pressure. Renin-Angiotensin-Aldosterone pathway. Decreased blood flow to the kidneys or decreased sodium levels stimulate the release of renin from the juxtaglomerular cells. Renin catalyzes the conversion of angiotensinogen to angiotensin I, which is then converted to angiotensin II by angiotensin-converting enzyme. Angiotensin II causes vasoconstriction, stimulates aldosterone release from the adrenal cortex, and triggers thirst, all leading to increased blood pressure and fluid retention. ADH, antidiuretic hormone. Pathway. Decreased blood volume or increased osmolality stimulates the release of ADH from the posterior pituitary gland. ADH acts on the kidneys to increase water reabsorption, leading to increased blood volume and blood pressure. ANP, Atrial Natriuretic Peptide. Pathway. Stretching of the atria in response to increased blood volume stimulates the release of ANP. ANP promotes vasodilation, increases sodium and water excretion by the kidneys, and inhibits the RAS, all contributing to decreased blood pressure. Stay updated with the latest in medicine. Subscribe to our channel for comprehensive videos covering the latest updates in the field.